Welcome all of you. Today I am going to start online lecture series. Myself, Assistant Professor Pravin Kale from Department of Zoology, SNJB's ACS College, Chandwad. I am going to start online lecture on the subject cell biology. B.Sc Zoology, subject code ZY-336. It is the third semester of B.Sc. In this today's lecture, chapter number 4, particular topic is Golgi complex. Golgi complex is the cell organelles, and this is your fourth chapter. In this chapter, some topics discuss with you. First is Golgi complex. In this particular topic, subtopic morphology of Golgi complex, then structure of Golgi complex, then function. So these three topics are discussed with you in this particular lecture. So first, Golgi complex. What is Golgi complex? Golgi complex is the cell organelles, and it is discovered by Golgi. Golgi is the Italian neurologist in 1898 who named it natural reticular apparatus. Then some botanists are referred to them as the dactysomes. This botanist name is a Baker, and this was discovered and referred to Golgi complex in 1951 and 1953. They are known as Golgi complex is lipochondria because of the presumed lipid contents observed in Golgi complex. And this structure similar to the Golgi complex have been found in plants, and they refer to them as dactysomes. The term Golgi complex has been preferred because of following reasons. Some reasons are there. First is electron microscopy demonstrated fairly discrete group of lamellar units. Second, Pronicito had earlier established a defined use of for dactysomes. Third, each dactysomes appear to the function in a membrane building product synthesis and assembly, and in the evolution of the products as an individual organelles. So this according to this botanist Baker in 1951, each dactysomes appears to the function in a membrane building product synthesis and assembly, and in the evolution of products as in individual organs. And electron microscopy demonstrated fairly discrete group of lamellar units. Then morphology of Golgi complex. The shape of Golgi is quite variable in a somatic animal cells. Even in the same cells, there are varying with a functional stage. In some cases, it occurs as the dense reticular of the anastomosing trabecules, while in others as an irregular laminated palque or hollow sphere united together. In nerve cells, it occurs as a reticulum of wide meshes around the nucleus. Golgi complex lies between the endoplasmic and plasma membrane. Endoplasmic reticulum and plasma membrane च्या मध्ये ते बघायला मिळतं. Then the shape and size of the Golgi complex. Size of Golgi complex is also variable, and it is small in a muscle cell but quite large in a nerve cells and glands. The shape of Golgi is quite variable in a somatic animal cell, even in the same cell. The number of Golgi apparatus per cell is also variable. Then the position of Golgi complex is relatively fixed or each cell type for each cell type. It is the morphology of Golgi complex. Position of Golgi complex, it is present between the nucleus and plasma membrane. Shape of Golgi complex, shape is variable in plant cell, even in the same cell shape is also variable. Then size is small in a muscle cell, large in a nerve cell. Then number is variable per cell. Some cells having single apparatus are there, and some cell having hundreds apparatus are present of Golgi complex. Then structure of Golgi complex. The first Golgi complex is proposed by Dalton and Flexin in 1954 by using electron microscope. They describe the Golgi complex in the epidermis of the rat as consisting of the three components: flattened sac or cisternae and large vacuoles. Flattened sacs or cisternae, large vacuoles, and small granules. These three components consist to the Golgi complex. All membranous structure and are characterized by absence of ribosomes. They are smooth membrane. Without ribosomes, membrane of Golgi complex is known as smooth membrane. Then element in a GC means element in Golgi complex. They are contain cisternae or lamella and vesicles. These are the elements present in Golgi complex. Cisternae or lamella are most constant element of the Golgi complex, and they are consist of flattened parallel sacs situated one upon the other and to form stacks. The number of cisternae in the stack varies from species to species. The number of cisternae in stack is about 4 to 5 in most animal and plant cells. In the euglena, the number may go up to the 20. Then vesicles. The membrane of Golgi complex are dynamic equilibrium, and they are continually receiving lamellae through budding of vesicles from the smooth endoplasmic reticulum and losing membrane through the formation of secretory vesicles. In the few cases, dense material has been found between the cisternae of Golgi complex and has been called intercisternal material. What is intercisternal material? In the few cases, a dense material has been found between the cisternae of Golgi complex and has been called intercisternal material. Vesicles, the second element of Golgi complex is vesicles. Vesicles are small vesicles are 400 to 800 Armstrong in diameter, and they are intimately associated with the cisternae. The small vesicles arise from cisternae by budding or pinching off. The large vacuoles are clear and generally lie at the edges of the Golgi complex, and they are represent modified and expanded cisternae, in which the two membranes have becomes widely separated and vacuolar space enlarged. They contain clear material which reduce the metallic salts. So these are the structure of Golgi complex. Then next is the chemical composition. Chemical composition of Golgi complex. The Golgi complex, it consists about the 60% of protein and 40% of lipid molecules. Morphology Golgi complex lies between endoplasmic reticulum and plasma membrane. The three membrane being linked by the transitional structure by the secretory granules. Golgi complex membrane are intermediate state differentiated between endoplasmic reticulum membrane and plasma membrane. Generally, it consists about 60% protein and 40% lipid molecules. Some proteins being common to those found endoplasmic reticulum but having fewer protein bands. And these bands is very low level of RNA, DNA and polysaccharides have been noted in some Golgi functions. Noted means observe some functions are there. Natural lipid fractions, mainly cholesterol, ester and triglycerides. Then the enzyme concentration in the Golgi fraction comprise thymine, pyrophosphatase and several glycosyl transferases. And it is the chemical composition of Golgi complex. The next functions of Golgi complex. Golgi complex show the different types of functions in a cell. Golgi and cell secretion or role in a secretion of Golgi complex. There are definite evidence which suggest relationship between Golgi complex and cell secretion, which was postulated by the Kale. Kale is the scientist name, and this is postulated in 1914. The Golgi and cell secretion, they are very fact that the Golgi are very well developed in all the cells exhibiting high secretion activity, clearly suggests that they play main role in the formation of secretion of granules. Golgi and सेल secretionचा main role काय आहे? The Golgi are very well developed in all cells exhibiting high secretory activity and clearly suggest that they play main role in the formation of secretion of granules. Many electron microscope studies show that during phytogenesis in oocytes, Yolk products first becomes visible in a cisternae of Golgi complex. Oocytes and yolk product during the phytogenesis, and these are first visible in a cisternae of Golgi complex in the many electron microscopic studies. However, in these studies, it's not clear whether of a yolk actually synthesized by the Golgi complex or only pack them. In certain oocytes, the Golgi complex appears to give rise to the critical granules. The Golgi cisternae may be pinched off to form small secretory granules, and these may then fuse to form large granules. Cisternae are completely filled by the secretory products. Second function of Golgi complex is the role in a protein secretion. They are important role play in a protein secretion. In the pancreatic exocrine cells, the distribution of secretory proteins means digestive enzymes. Secretory protein also known as the digestive enzymes. It has been investigated by a Sakevich and Palade in 1962. First, they are according to Sakevich and Palade, proteins are formed on the ribosomal attachment to the endoplasmic reticulum. Then these nascent proteins are then transferred into the endoplasmic reticulum. Third, Golgi complex form here. They go the Golgi complex. There is no morphological evidence that the endoplasmic reticulum may bud of vesicles which travel toward the Golgi complex, and the vesicles may be fused with the membrane of Golgi cisternae and release their content into the latter. Then bud of this Golgi cisternae and in the then secretion. Then next function is synthesis of carbohydrates. Golgi also synthesize the carbohydrates, means sugars. First, in the plant cells, it has been demonstrated to produce pectic materials which are enclosed in vesicles moves to cell wall region. And here, vesicle membrane fuse with the plasma membrane and vessels contains the accumulate between the cell wall and the plasma membrane. Then carbohydrate synthesis, it is the similar observation have been made in the formation of new cell plate at the time of cell division in plants. Abundant dictyosomes cut off vesicles which move to the cell plate region and they are fused together, releasing the constituent of a new cell plate. This is the synthesis of carbohydrates. It's the function of Golgi complex. Then enzymes. Golgi complex secretes different type of enzymes content, including acid, have often been demonstrated to be active in a dictyosomes. The most characteristic enzymes associated with the Golgi membrane, and those are linking sugars with proteins, and to form glucoproteins. Enzyme associated with Golgi membrane are those linking sugars with proteins and to form glucoproteins. For example, mucopolysaccharides. Mucopolysaccharides used by the hydra for attaching its base to the substratum. Next function is polysaccharide synthesis of polysaccharides. Nutra and Lebold carried out the autobiographical studies. Golgi produce pectic substances and different polysaccharides are there. Then next, it takes an active part in the incorporation of substance from the environment. Precursor enter the globulate cells from the capillaries of vascular systems The amino acid are synthesis into the protein on the ribosomes of endoplasmic reticulum. The proteins are then transferred to the cisternae of Golgi complex. The next function is the pigment formation. Golgi complex also play in important role pigment formation also. Then last one, formation of acrosome during the spermatogenesis. During the maturation of sperm, the Golgi plays an important role in the formation of acrosomes. In the early stage, Golgi appears as the spherical bodies comprising the cisternae and arranged in a parallel stalk and numerous small vesicles. These are always pinch off from the cisternae. As the development proceeds, the Golgi becomes irregular in shape, and large vacuoles are formed by the dilation of cisternae sacs. In the central and these large vacuoles are present dense granules and precursomal granules. Dense and precursomal granules. These granules seem to be enlarging vacuoles, and these vacuoles, along with the granules, approach the anterior fold of the nuclear membrane, constituting acrosomal granules. With the elongation of spermatids, the acrosomal vesicles spread over the nuclear surface and finally collapse with the nuclear membrane. See in this diagram, formation of acrosomal during the spermatogenesis. In early stage, Golgi appears as the spherical body. See in diagram number first. Then comprising cisternae arranged the parallel stalk and numerous small vesicles. Then nuclear consisting membrane acrosomal granules with the elongation of the spermatid, and acrosomal vesicles spread over the nuclear surface and finally collapse the nuclear membrane. See in diagram number 2, they collapse their nuclear membrane and forming a cap material. Forming a cap formation होते हैं diagram number 3 मध्ये. Flow wise respectively first, second, third and fourth. Last is the sperm. Becomes the acrosomal, which lies at the apex of the nucleus and apparently comprise certain enzymes involved in this process of fertilization. See in this diagram, Golgi rest, then their nuclear membrane collapse and formation of the sperm. Sperm region, head, middle face, and the last one is the tail region. So pigment formation in many mammalian tumor and cancer cells, the Golgi complex has been described as the site of origin of pigment granules, and these granules are pigment granules, also known as melanin. The Golgi complex has also been associated with the pigment formation in renal pigment epithelium of the chick embryo, in the test cell of ovaries and certain tunicates, and in the oocytes of the salamander. So remember some points are there. Morphologically, Golgi complex lies between the endoplasmic reticulum and plasma membrane. The membrane of Golgi complex, endoplasmic reticulum and plasma membrane are linked by the transitional structure and by the secretory granules. Then the position of Golgi is relatively fixed for each cell type. The Golgi complex first discovered by the Golgi in 1898. The Golgi apparatus is a system of roughly parallel interconnecting flattened sacs situated close to the endoplasmic reticulum but physiologically separate from them. It helps in a secretion, cell secretion, protein secretion, synthesis of carbohydrates and pigment formation. Okay, thank you for watching. Next lecture on lysosomes. नेक्स्ट लेक्चर बघण्यासाठी व्हिडिओला लाईक करा, सबस्क्राईब करा, ज्याच्याने तुम्हाला नेक्स्ट लेक्चर इझिली व्ह्यू होईल. त्यामुळे आत्ताच सबस्क्राईब करा आणि व्हिडिओ शेअर करायला विसरू नका. थँक्यू.